A very good morning to everyone, and I welcome you all to the first lecture of the 1 week international e-workshop on bioinformatics. Before starting the session, we have few guidelines for the participants to ensure the easier understanding and management of the lecture. First of all, we request all the participants to be ready with their desktop or laptops for hands-on session. We will take the attendance for each session. The attendance sheet will be available during the lecture in the chat box. Minimum 80% attendance is essential for receiving the e-certificate. Also, at the end of the workshop, there will be a feedback form which needs to be submitted for receiving the e-certificate. Please fill your name and email addresses carefully. We request you to post the queries or doubts of the lecture in the chat box. We would try to address most of your queries after the respective lecture is over. Thank you everyone for the cooperation. It is time for me to make you familiar with the personality without whose vision and sincere efforts this workshop would not have been the reality. The lady herself, the coordinator of the workshop, Dr. Yasha Hasija. Dr. Yasha Hasija is currently working as an associate professor at Department of Biotechnology and associate dean alumni affairs at Delhi Technological University. Ma'am has research experience of more than 10 years in genome informatics, human disease genomics, genome annotation, integration of genome scale data for systems biology, personalized genomics, and machine learning applications in healthcare. In her career down the lane, she has published more than 100 research papers in national and international journals and conferences of repute, and 10 book chapters. She is also the project investigator of several sponsored research projects from Government of India, agencies including DSTSERB, CSIR-OSDD, and DBT. She has been awarded several prestigious awards, including the Commendable Research Award 2018 at Delhi Technological University, along with the Nomenclature Prize in Human Genome Meeting in France. She is also in the editorial board of various journals, including PLOS ONE, BMC Research Notes, and the American Journal of the Medical Sciences. Thank you, ma'am, for lighting us path towards this direction. To onset our first session of the first day, may I request now Dr. Yasha Hasija, Associate Professor and Coordinator of the workshop, to introduce our first speaker, Dr. Vinod Scaria. Ma'am, please.
Thanks a lot, Neha. A very good morning, everyone, and a very warm welcome to the first hands-on session in this international e-workshop on bioinformatics. As they say, we do our best when we do it together. So I believe this workshop is one step in the right direction to furnish synergy between all of us for one goal we all want to achieve. Learning must never stop. And we're all here to learn and apply informatics in this era of big data in biology. I'm glad to introduce Dr. Vinod Scaria, Senior Scientist at CSIR-IGIB, Delhi, India. Dr. Scaria is a clinician turned computational biologist. He is involved in creating novel methods and resources for analysis and annotation of genomes and understanding the functional impact of genomic variations. He has been part of collaborative genomics projects aimed at understanding the Asian genome diversity. He has also been a part of the whole genome sequencing and analysis projects, including the Indian, Sri Lankan, and Malaysian genome projects, and is a member of the Hugo Pan-Asian Population Genomics Initiative Task Force. He is also the co-founder of the Genomics for Understanding Rare Diseases India Alliance Network, or Guardian, which is one of the largest networks of clinicians and researchers in India working on rare genetic diseases. He has adopted novel and creative strategies, such as the use of social media and the participation of a large number of undergraduate students in collaborative projects to accelerate genome annotation and co-creation resources for genome annotation. Dr. Vinod did his undergraduate medical education from Calicut Medical College, University of Calicut, and PhD in computational biology from University of Pune. Vinod has over 90 publications in international peer-reviewed journals, a book and 2 book chapters to his credit. He is also in the editorial board of PLOS ONE, PLJ, Journal of Translational Medicine, Journal of Orthopedics, and International Journal of Rheumatic Diseases. He is a recipient of CSIR Young Scientist Award for Biological Sciences in 2012 and a Kavli Frontiers of Science Fellow of US National Academy of Sciences in 2015. I invite Dr. Scaria to take over his session on what did we learn from the genomes of SARS-CoV-2 from India. Dr. Vinod, please.
Thank you, Dr. Yasha, for the nice introduction. It's a pleasure to be here and talk to you all. Just a minute, let me just put the slides on. I hope the slides are visible.
Yes, yeah.
So I've planned the talk in such a way that we would have a lecture for almost 45 minutes to an hour, which could be followed by as many questions as I could take, and a short sort of demonstration of the resources that we have created to look at SARS genomes and understand them much better. So before I start my talk, let me first introduce our lab. We work in the area of genome informatics, which necessarily means that we look at genomes of organisms on the computer and try to make quite some interesting guesses about how they are functional and how they are organized. We work on a variety of organisms. Humans, of course, are of great interest to us, but apart from that, we also work on model system genomes, including zebrafish, mouse, as well as rat, trying to understand how their genomes are organized and how we can draw parallels between the human as well as these model system genomes. The other part of my lab works at the interface of human genomics and medicine, where we try to incorporate the findings in human genomics to help people understand very specifically diseases. And to be exact, we work on rare genetic diseases, which are familiar in occurrence and quite prevalent across the country. A small part of my lab also works in the area of pathogen genomics, where we look at the pathogen genomes. We used to work on Mycobacterium tuberculosis before, and very recently with the COVID epidemic, we initiated a work on COVID. And that's essentially what I'm going to talk to you over the next half an hour to 45 minutes. You could also find more information about our lab and what we do on our lab website. Now, I'm sure this epidemic has been quite remarkable in many ways, because one tiny virus, which emerged almost a year ago in Wuhan in China, with a very small genome of around 30,000 base pairs, has actually wreaked havoc in all our lives, not just professionally, but also in many aspects of the fabric of the society. Now, all that we know about this virus is thanks to genomics. And that's also what the point I'm going to make and put across is that this epidemic has been quite remarkable even in the field of genomics. And one reason why I think genomics has really revolutionized how we look at epidemics is because it has traditionally been used to look at genomes of larger organisms, like, for example, the human genome, but could be quickly repurposed to look at genomes of viruses. And more importantly than all of it was to revolutionize the way microbiology is practiced. Now, quite remarkable in this epidemic has been the approach to be able to sequence viral genomes from human samples without the need of traditional microbiology. Now, when I say without the need of traditional microbiology, I mean to say that traditional microbiology has always been reliant upon approaches or methods to culture, to purify, or to enrich sets of microorganisms, whether it is bacteria or whether it is virus. The identification of the pathogen typically was preceded by an approach to enrich or purify the organism. Now, that is what has really changed in this particular epidemic. It is not that these technologies were new, but technologies were quite old, but has been used to application in such a situation. Now, the basis of all of this application is one, the throughput of sequencing that you could afford to have, and second, the computational approaches where you could reconstruct genomes from mixed samples of organisms. Now, The throughput, of course, you know about next generation sequencing, which has significantly improved the throughput of genome sequencing. And this technology is almost 15 years old. Sorry for that. And of course, much more importantly has been algorithms to be able to look at these samples, look at these pieces of DNA, and we assemble them into organisms or complete genomes of organisms and be able to identify the organism. Now, the virus as we know today was thanks to the genome sequence, which was made available early in the epidemic by researchers in China. And this paper essentially exemplifies the approach that I sort of mentioned. And this approach took the patient sample, bronchoalveolar lavage, which is essentially a sample aspirated from the bronchi of individuals. And the sample directly went into a total RNA extraction, ribosomal depletion, and sequencing. And this is not typically what epidemics or how epidemics have been investigated previously. Typically, epidemics would be investigated by isolating the virus, culturing the virus, and being able to understand the genome sequence. So in many ways, previous epidemics, the genomes, followed the epidemic by a few months, if in some cases a few years, because of approaches that are required to purify the virus. But contrary to all previous epidemics, this epidemic actually saw the viral genome coming in first, probably even coinciding with the identification of the causative organism. Now, what happens after RNA sequencing is a quite elaborate process of genome analysis and interpretation. There are few protocols which are available online, which students could actually follow. I'll delve deep into that in the practice session. Now, the other question that you need to, or the other point that you need to put in mind is that the virus is evolving. It is not just SARS-CoV-2 is evolving, but all viruses, all organisms are evolving. The only difference between other organisms is the speed of substitutions or the speed of mutations that happen in an organism. Now, as we know, SARS-CoV-2 is an RNA virus, which has a much higher rate of evolution or rate of substitutions. And you need to keep in mind that approximately one substitution occurs in the viral genome once in every week or so. So that is what you need to keep in mind, because this is something that is going to be the essence of all that we do in genomics. Now, to exemplify this point, you could actually put in factual information. The first Wuhan genome, which was the SARS-CoV-2 virus from Wuhan market, was made available early. This virus was isolated in December 26th. And as you see here, the genome from another virus from Jan 8th from Wuhan was also sequenced, and you would see that it was different from the original strain or the original isolate by one nucleotide. And of course, if you look further, there have been additional variants that have occurred in viruses isolated from different parts of China. And for example, this case were from Guangzhou in China. Now, this is what you need to keep in mind as a virus mutates, and this mutation is pretty much at a constant rate. And therefore, you can use this information to understand a lot of things about the virus. Now, where do you find these viruses? There's been a large number of viruses that has been sequenced and are made available over the last 1 year. Today, we have crossed over 200,000 genomes of SARS-CoV-2 available in multiple different databases across the world. And two large databases that you'd really look for is one, the NCBI GenBank, and the other one is called the GISAID. And of course, there are repertoires or repositories of viral genomic data in many other country-wide programs, like, for example, the COG UK consortium, which has sequenced approximately around, I believe, 100,000 odd genomes of this 200,000. Now, that's one great part of genomics, that a lot of data is available in open domain. And thanks to all the researchers who made this data available and have shared in the spirit of understanding the pandemic better and faster. Now, one of the resources that you will also see in the later practice session is this resource that we developed very early in the pandemic. This was developed in April with the idea that there was no search engine to be able to look at genomes of SARS-CoV-2. So this search engine essentially indexes genomes from across different databases, and you can search using multiple parameters, like for example, the data source, the country, the originating lab, the submitter, the submission ID, so on and so forth. And therefore, you can access the right information or the right data set that you want to go back and do the analysis. Like for example, you could immediately search for genomes from India, or you can search for genomes from Delhi, and so on and so forth. And therefore, this becomes a very unique resource to access genomes for your analysis. Now, I sort of mentioned this before, but sort of reiterating it in a different context. The virus, as I said, accumulates a mutation once in approximately around 7 to 10 days. Now, what you should remember here is that when the virus accumulates a mutation, it passes on that mutation forward, and that forms a chain, right? And for example, in this particular case, you have a mutation that is accumulated. I call it mutation 1. Then you have mutation 2, and mutation 2 could accumulate 1 to 4 in the fourth position, or you could accumulate a mutation at third position, and that becomes 1 to 3. And 1 to 3 can accumulate another mutation, becomes 1 to 3, 5, and so on and so forth. So it essentially forms a chain of events. Now, if the transmission of the virus was uniform, that means the individual transmitted to just one another individual, and that individual to one another individual, at any point in time, you are going to have just one set of mutations that accumulate. But now assume that at some point of time, one individual transmitted the virus to two individuals, and therefore that's the individual what I call who has transmitted to two individuals. So for sake, I'll say 1, 2, and transmitted it into 1, 2, 4 and 1, 2, 3. Each of them accumulated one mutation further in 7 days. And then therefore, if you look at a time point in space, and this is how you could have a time point in space, the proportions of each of these chains would essentially be the same. If the transmission was uniform, then the proportions of these chains would also be the same. But that is not in reality what happens. In reality, what happens is that there are some individuals whom we call as super spreaders who can transmit to multiple or large number of individuals. And for the sake, let us call 1 to 4 as a super spreader who has transmitted to a large number of individuals. So if you look at the genomes at a time point, then you would see a large number of viruses with the 1 to 4 mutations compared to the 1 to 3 or 1 to 3, 5 or any other mutations. Okay. So this is how a particular clade or a genetic lineage emerges out of a transmission chain. And 1 to 4 is what we call as a point of emergence, or that's an individual who has spread to a significantly large number of individuals, or a disproportionately large number of individuals, which now reflects on a time scale sequencing as a large proportion of that particular genetic lineage. Now, this is essentially what we call as genetic lineages or genetic clades. And to make it very simple, so there are clades or genetic lineages that can be drawn on a phylogenetic tree. We don't call them strains of the virus because they're in fact only genetic lineages of the virus. And they have much lesser number of genetic variations compared to strains of virus. So then therefore, we need to call them scientifically as genetic lineages or genetic clades rather than strains of the virus. So there is SARS-CoV-2, which is a strain of the virus, significantly different from the MERS or the SARS-CoV, which was the first and the second outbreaks of SARS-CoV. This is SARS-CoV-2, which signifies it different from the SARS-CoV-1 or the first SARS epidemic. And everything in SARS-CoV-2 is not a strain, but only a genetic lineage. Now, you can plot genetic lineages of virus from across the world. And this is one resource that you could access. It is called NextStrain, quite popular in the genomics community. So if you look at this map of genetic lineages across the world, you would see that one lineage of the virus, right here in blue, essentially predominates all the other lineages of the virus, which you see here in yellow, orange, red, blue, or green. Now, this is what we call as a clade of the virus, and this large clade is what we call as a A2A clade of the virus. So to be precise, globally there are around 10 genetic lineages of the virus, or 10 clades of the virus, which are essentially two superclades, the A1 or the A superclade and the B superclade. The B superclade are essentially isolates from China, early in origin, and the A are essentially isolates which emerged out from the West, mostly Europe and adjoining areas, and has spread across the world. Okay. So, in summary, there are 10 genetic lineages. In early points in time, these genetic lineages signified from which country did this virus come from. So, in the epidemic, the B clades came from Wuhan in China. The A clades or the A2A clades came from Europe, so on and so forth. But going further, you lose the information about where did this come from, because you could have internal transmission of the virus in a particular community or in a particular population. Okay. Now, just to reiterate this point, that there are mutations, and using these mutations, you can now use the chain of events, or looking at the chain of mutations that happen in a virus, you can allow molecular contact tracing or ancestry tracing. Now, coming back to India, the first report of SARS-CoV-2 in India was in 30th of January 2020 in a set of individuals who had a travel history from Wuhan in China. And now in December, we have almost 9 million, 9.7 million cases with a significantly large number of deaths. Now, you would ask this question, obviously, did all the viruses come from Wuhan in China directly into India, or did it come from different parts of the world into India? But nevertheless, one thing is sure, that practically all viruses that you see today in India has come from travelers who have traveled from across the globe. Now, to put numbers into perspective, there are approximately today over 4,000 genomes of SARS-CoV-2 deposited from a large number of institutes from across the country and spreading practically almost all states and union territories. I'm sure Dr. Anurag Agarwal sort of mentioned about this. Now, one way of sequencing viruses was to also use the high throughput technology, where you can sequence to detect, as in to identify the virus, and also use the genetic information to build genetic epidemiology. Now, this is one such approach where we have standardized the clinical application of clinical samples which can be sequenced to detect whether they have SARS-CoV-2 or not, and also use the genetic information to draw contact traces or understand where did these viruses come from. And this was developed in the lab of Dr. Sridhar Chivasubbu, who is my close colleague and collaborator, with a lot of young enthusiastic people who have contributed to this entire effort. Now, in summary, how this happens is through a surveillance program we sort of put together. Typically in the surveillance program, what we offer individuals who are part of the surveillance is a tube with a swab. They typically take their own samples and deposit the tube back with an ID. Now, this ID can be checked on a mobile app to understand whether you're positive for the virus or you don't have the virus. And how do we do this? We do this by sequencing. As any The only difference is that this is high throughput. That means you can generate approximately around 3,000 samples worth of information in 12 hours using a sequencer, which is a Novosec 6000 system, which is typically used to sequence human genomes. Now, what happens behind the doors is a quite elaborate system where you could take samples, isolate RNA, and then plate them into 96 well plates along with control RNA and sequence them in high throughput. And once you have sequenced, assemble back the samples because they are barcoded. Each sample has a barcode. You can deconvolute the barcodes. You can assemble the genomes. You can call the variants and release the reports. And this entire activity takes 24 hours from sample to report. And this entire thing is sort of automated so that you can put in a sample at one end of the chain, and at the end of 24 hours, you should be able to get a report on whether you are positive for SARS-CoV-2 or not. And we use this approach to understand the viral epidemic in multiple southern states, mostly Kerala, Tamil Nadu, Andhra Pradesh, Karnataka, Orissa, and Uttar Pradesh. And Uttar Pradesh is mostly the NCR, Noida, Greater Noida, and adjoining areas. Now, approximately half of all genomes that are made available in India is through this approach called the COVID-Seq, where you could detect the samples and, of course, offer them a report early in the epidemic, and also use that information to be able to build phylogenetic trees and also quite importantly, guesses about how did this virus emerge and how did this virus get transmitted across the country. Now, if you look at the phylogenetic tree of India, and this is from, I believe, from August, what you see in India is out of the 10 distinct clades or genetic lineages which are present across the world, we have 6 of them. And apart from the 6, we have one distinct cluster here marked in violet, which seems to be quite predominant in India and not found predominantly in any other country across the world. And since this was a distinct cluster, we sort of named it as the I or the A3I clade. I'll sort of explain that a bit of slides from here. You would also see the initial genomes from Kerala, the 2 initial genomes from Kerala, which was deposited by the National Institute of Virology, which belongs to the B superclade of the family, which sort of confirms its origins from China. And the A3I essentially is very close to the A3 cluster here in green. You see a small speck here in green. And this green samples essentially are travelers into India from Iran. But nevertheless, our analysis suggests that it didn't emerge in India, but it emerged elsewhere, because as you see here, the branch point precedes the samples that has come into India from the A3 clade. But what is also significant to note is that a significantly large number of the viruses today circulating in the country is belonging to the A2A clade, which essentially arose in Europe, and therefore we believe that a significant number of influx of SARS-CoV-2 into India came from travelers who have traveled from Europe as well as the Americas. Now, each of these clades are also marked by specific mutations. In this particular case of the IA3I clade, there are 4 genetic mutations which sort of mark this virus, contrary to the A2A clade, which is essentially marked by 2 genetic mutations, A3 by another 2 genetic mutations, B and B4 by 2 and 1 genetic mutations, so on and so forth. So in other words, what you look in the genome to understand the clades are essentially just a handful of genetic variations, and these are essentially the fingerprints of the clades. And if you understand what is a mutation that you have, you can immediately map them into a genetic lineage or a genetic clade. So this map or information is quite useful to understand, one, where did this virus come from in the initial part of the picture, and second, how did it diverge out into very different clades. As you see here, the B and B4 has one common mutation, which is also shared with B1, and B4 and B1 have additional one each mutations, which are distinctly different from the common mutations in the B superclade. So this is the 1, 2, 3, 1, 2, 3, 5 sort of chain that I sort of mentioned. So you know from here that the initial superclade was B, which diverged into B1 and B4. And of course, if you know the rate of mutations, you can also date when did this occur and where did this occur. Now, coming back to the IA3I mutation, we went back and asked this question across the country: where does these clades occur? And what you see here is the map of the predominant clades. The predominant clades are A2A and the IA3I. And this is sometime June, July in India. And what you see here is the distribution of the different clades are quite distinctly different in different parts of the country. Now, if you look at Gujarat, which has a large number of genomes that were sequenced from the GBRC, you would see here that a significant number of genomes were from the A2A clade. But now, for example, if you look at Delhi, or if you look at, for example, Telangana or Tamil Nadu, you'd see a significant number of genomes belong to the IA3I clade of the virus. And of course, there are multiple different states which has distinctly different proportions of viruses uh, or the clade of viruses. Now, what we also know is that the IA3I clade seems to have originated from a point event or a super spreader event, and then diverged out into multiple different states, as you see here in the second panel. It seems to have originated from a few individuals, one or few individuals, and then have spread across to different parts of the country. And you could also look at whether this genetic clade of the virus mutates distinctly different, because you have time series data for this clade spanning 3 to 4 months. And what we understood from here is that compared to the A2A clade of the virus, which is approximately 1.42 into 10 to the power minus 3 substitutions per base per year, which approximately translates to around 3.7 per month, or approximately 1 per week. The IA3I has a significantly lower rate of mutation, approximately 1 3. Now, what does it mean? It means that the virus is probably in some ways defective in accumulating mutations, and accumulating mutations is essentially a survival strategy, an evolutionary strategy for a virus. So we sort of predicted at that point of time that this virus probably is going to get eliminated because it doesn't have a survival advantage or a fitness advantage. And that's exactly what we see in the data. The violet is the IA3I, and over time, as you see here, and the x-axis is the weeks. And what you see here is over time, the IA3I clade has sort of vanished from the repertoire of viruses that are circulating in the population, and overwhelmingly has been taken over by the A2A clade of the virus. And this is the top panel is the cumulative data, and the bottom panel is essentially the data from different states. Now, there are multiple reasons that we could attribute to this. One is, of course, that it has a lower mutation rate. And apart from that, there have been also early efforts to control, including multiple rounds of lockdown and restrictions of interstate travel, which typically would have culminated in this clade of the virus sort of vanishing from the scene. Now, of the states, the IA3I clade has been found largely in Delhi and Telangana. And of course, apart from these two clades, the A2A and IA3I, there are some minor proportions of other clades across the country, which includes the B1 and the B4, and of course the A3, which was early found from Ladakh, from travelers from Iran. But all of these seems to have sort of faded out and eventually taken over by the A2A clade of the virus. Now, I sort of said that we looked at the genetic epidemiology of multiple different states. And one of the states we were really interested in looking at was Kerala because of multiple reasons. One, it was a state which seemed to have controlled COVID quite effectively early in the epidemic. And it was a state which saw the first sort of SARS-CoV-2 in an individual traveling from Wuhan. Third, it has a very large expatriate population, mostly in the Middle East, Europe, and Americas, who are also traveling back into the state and had a strict quarantine measure. And fourth, it was adjoining multiple other states, like, for example, Tamil Nadu and Karnataka, which saw early outbreaks of SARS-CoV-2. So we were interested in understanding if you now do a cross-sectional analysis of some samples from Kerala, can we really understand where did this virus come from? And what we found was quite remarkable, that practically all the viruses that we have identified were from the A2A clade of the virus, and absolutely none from the B clade or the IA3I clade. Now, I said before, the B clade are essentially people who traveled out of China early in the epidemic, and the A2A are essentially people who traveled out of Europe. So we asked this question. Now, if you map these contact networks based on the haplotypes or the chains of genetic mutations, what we get is three large clusters. We call them for convenience K1, K2, and K3. And apparently, none of these clusters seem to have a quite large inflow from Europe. That necessarily means that these viruses came into Kerala not from international travelers, but probably from travelers of nearby states. And as you see here, K1, K2, and K3 seems to have originated from nearby states, uh, uh, depending upon the root of the cluster or the nearest neighbor of the cluster. But nevertheless, these are small outbreaks that has occurred within the state early in the epidemic. And then therefore, you could understand not just how did the virus come into a particular region or a particular state, but also where and how did this virus spread in the community. So K1, K2, and K3 are essentially clusters of outbreak, which has happened within the state in many ways undetected, because RT-PCR will never tell you where did this virus come from. It will only tell you whether the virus is positive or negative. In at least one of these outbreaks, we were sort of able to go back and do an extensive contact tracing. And what we realized is that one of these outbreaks essentially came from a hospital where apparently a patient was admitted unknowingly, who was tested negative for COVID eventually, but had transmitted the virus to a large number of people. Now, if you ask this question again now, so this is a map from around November. What you see here is the proportions of the clades have now dramatically changed. Delhi still have a significant number of IA3I, approximately, I believe, around 27%. But in other states, the numbers of the IA3I have really come down. And the bottom panel is essentially the time scale map of the different clades across the different time points. And overwhelmingly, all the clades have been replaced by the A2A, which is the light blue color, all over, except for a certain number of B4 clades, which is now present in West Bengal. And you can use this information to also build maps of contact tracing. And this is essentially important because there are a large number of people who have traveled across states in different points in time. The migrant labor who traveled across from city to city during the early lockdown, and of course travelers who traveled across different states late in the epidemic after the lockdowns have been lifted. So you can actually sort of play a video or a timescale video on how these viruses have spread across from state to state or city to city on this map that comes in the exercise section later. And you could now build such haplotype maps for all the genomes, and this is for approximately 4,000 odd genomes from India. And now you can understand when and where did these outbreaks occur. As you see here, the large blobs or large outbreaks which have occurred in Gujarat, the large outbreaks which have occurred in Kerala, the large outbreaks which had occurred in Mumbai, in Delhi, and so on and so forth. So in other words, retrospectively, you can now understand where did these outbreaks occur, how did these outbreaks occur, when did these outbreaks occur, and how did it spread across to different states. So this is two implications. One, it can understand how did the virus spread. So in the future epidemics, you exactly know how to curb the spread of the virus. Second, in many cases, there have been interstate spread of viruses. So this allows you to go back and sort of curb the set of people who are likely to transmit this virus going forward. And all these data sets are available in this resource called INDICOV. I'll sort of briefly mention this resource later in my talk. The other application of genomics is to understand what we call as reinfections. I'm sure you would have seen a lot of cases of reinfections reported in press. Reinfections essentially means an individual who have caught SARS-CoV-2 infection twice in this epidemic. Now, that necessarily means that you should have an individual tested twice for SARS-CoV-2, spread apart by a few months, and it needs to be differentiated from a condition called as reactivation or persistent viral shedding. Now, persistent viral shedding essentially means that an individual tested positive for SARS-CoV-2, and typically an individual becomes negative in approximately 7 to 14 days, or the viral shedding stops at 7 to 14 days. But in some minority of individuals, these individuals continue spreading the virus for months altogether. The other condition is called reactivation, where an individual would have got SARS-CoV-2 and eventually tested negative for PCR. And after a point in time, the virus reactivates and re-emerges out into a second infection, which is extremely rare. And as per literature, this is seen only in cases of immunosuppression. Now, early in the epidemic, we have been working with the Government Institute of Medical Sciences, or GIMS, in Greater Noida, a government medical college catering to the large population in that area. And what we have sort of helped them is to put together a surveillance program for healthcare workers. And all healthcare workers who are in COVID duties were tested on a very regular basis for SARS-CoV-2 using RT-PCR. And that's how we identified these two individuals who tested positive for RT-PCR on 2 episodes. And fortunately, during the early epidemic, they were also tested negative for RT-PCR. Now, how do we differentiate between reactivation, persistent viral shedding, and reinfection? The only way to differentiate is to show that the viruses which are there in 2 infections are from different genetic lineages or have different mutations which are not compatible with the same genetic lineage. And that's exactly what we showed. And this is essentially the virus from the episode 1 and episode 2 for the 2 individuals named I1 and I2. And what we show here is that the viruses in the 2 infections have distinctly different genetic mutations which are incompatible with persistent viral shedding or viral reactivation. In persistent viral shedding or viral reactivation, you would see the same virus, probably different by 1 or 2 mutations. But in a case of reinfection, you would see distinctly different viruses in the 2 episodes. Now, of course, there are a very large number of cases of reinfections reported from across the world. Around 50 cases have been confirmed across the world. And probably in India, as per genomically confirmed, there are around 6 cases reported. And we are working on a very large number of cases from different states using genome sequencing. Now, is this the only application for genome sequencing? The answer is no. Genome sequencing can be also used for a variety of other things, like, for example, building better diagnostics, understanding immune epitopes, and understanding how do vaccines work, or more importantly, what are the conditions in which the vaccines will not work, using this information to build better vaccines, and of course, understand potentially functional genetic variations in the virus. I will not delve deep into all of these, but of course, you could find a number of reprints on these topics on my website. For the sake of time, I'll discuss one point on how to make better diagnostics using genome sequencing. Now, coming back to diagnostics, the method of choice for diagnosis of SARS-CoV-2 is a method which is called real-time PCR. And real-time PCR is based on the concept that you have oligonucleotide probes which can bind specifically to regions in the viral genome and amplify those regions. Now, therefore, you could also surmise that if there are genetic variations in these sites where the primers or the probes bind, then you could have ineffective binding and then therefore failure for the diagnostic test. So what do you do to understand such genetic variations? You need to first get the information of the primers and the probes. And that's not very easy because most of the companies do not disclose what are the primers and the probes which are used in their kits. So what do? You go back and ask these companies. There are around 500 odd companies today which are making diagnostic kits. So you mail them or ask for the literature, or if they have published the assay elsewhere, use that information to retrieve the primer and the probe information. And this was essentially done by a bunch of students from Bangalore who contributed their time during the lockdown to a Nobel course. And after you have sort of compiled this information, what you could do is to map genetic variations and ask these questions: are these genetic variations frequent in a particular population? And frequency cutoff sort of we have put in here is around 1%. Or in other words, if there is a genetic mutation in a primer or probe site and is more than 1% in the population and is going to affect the binding efficacy of the primer or the probe, you could calculate the efficacy or the free energy of binding using thermodynamic equations. Then you know that these genetic variations are likely to affect the efficacy of diagnostic reagents. Now we have a few genetic variants which affect the efficacy of some kits, mostly the N gene. This is also published and you could find the reprint out there. But more importantly, the genomic map also allows you to identify regions in the genome which are devoid of genetic variations, which are frequent in the population. And then therefore, you could use these genome regions to be able to design better diagnostics. And of course, for interested people, you could find a lot of more information on the COVID-19 Open Research and Resources website. And before I close, let me acknowledge a lot of people which made this possible, including people in my lab, a lot of volunteers which made this possible, fantastic clinical collaborators from across the country who made possible the COVID-Sec genetic epidemiology, Sridhar for most of the next generation sequencing, and fantastic collaborations with Divitej and Rakesh Mishra from CCMB. And of course, this is all reliant upon a lot of data across the world and across India were put together by a large number of resources in public domain. I think I'll stop there and take questions. But before that, for students who are interested in participating more on open research efforts, there's this program that we are currently running, and feel free to take a look and participate. I'll take questions and then move on to the second part of the lecture.
Thank you, sir, for this part. And I have the participants' questions ready. So, starting with the question from Miss Suviksha. So, she asked that how have the outcomes of research conducted for SARS-CoV-1 and MERS helped in the genomic research of SARS-CoV-2?
So, of course, the genomes of the initial SARS viruses were essentially extremely useful because the early diagnostics that were built for SARS-CoV-2 were essentially built on the concept that there are regions which are conserved between the different SARS viruses, and the regions which are distinctly different in SARS-CoV-2 and not in the earlier coronavirus epidemics. This essentially formed the sort of basis for the screening test and the confirmation test. As you would have imagined, earlier RT-PCR test in India or across the world used two tests. One was a screening test and the other one was a confirmatory test. The screening test was typically based on the E or the N gene, and the confirmatory test was based on the RDRP, or otherwise known as the ORF1ab gene. Now, this information of what is specific to coronaviruses and what is distinctly different in SARS-CoV-2 from other coronaviruses is essentially the basis of all diagnostics that we build and use.
Okay. Thank you, sir. So, next question we have from Miss Tina, and she's asking that is it possible that the coronavirus, which is responsible for any mutation in the infected individual, like it may later on affect on the next generation also?
I didn't get your point. Could you repeat that question once more?
Yes. So that is it possible the coronavirus, which is responsible for mutation in the infected individual now, and it can affect on the next generation, like after the vaccine has been, given to it?
Okay, so typically when a virus infects you, you have a period where you have viremia, and then your body typically takes care of the virus, and then the virus shedding stops. So by that time, you're typically immune to the virus. But of course, by contact, you would have spread the virus to somebody else, and that's how the virus spreads across the country. Now, your question is, if I have a virus and if it mutates, does it affect me, or does it affect the vaccine efficacy?
Yes, sir.
Probably no, because in the immediate sort of time period, these virus mutations are not going to be very what you call problematic. But yes, having said that, there we have come to notice some genetic mutations which can now escape the immune mechanism, and we propose that that's one of the mechanisms where the reinfections occur.
Okay. Okay, thank you, sir. Sir, but many of the participants are also asking about the tools which you have used to draw these infographics and to create these haplotypes map. So, can you give them a brief idea about the tools? What kind of tools have you used for this?
Okay, so typically there are three kinds of tools that you need to understand to essentially handle the viral genomic information. The first is essentially the basics, which is assembly, right? So you could assemble the genomes from the raw reads. So we have an extensive protocol for viral genome assembly. I will sort of mention that in my next session. The second part is, once you have the assembled virus, you need to do a phylogenetic analysis. The multiple ways of doing the phylogenetic analysis, and one that we follow is what is followed by NextStream. There's also an elaborate protocol that we have put together, which students can follow to be able to reconstruct the phylogenetic information. The third part of it is essentially looking at genetic variants and interpreting them in respect to the epitopes, in respect to the diagnostic kits, in respect to the functionality of the variants. And that is where the third resource, where we call as IndiCove, compiles all this information and makes it available as a searchable format. So these are the three distinct sets of tools that you will need to look at whenever you come across a genomic data for SARS-CoV-2. And my next session will I'll not explain each of these items, but I will sort of show you how you can access these resources online and be able to do what you want to do.
Okay. Thank you, sir. So my next question is about the vaccines. Would the vaccine have similar result for all the different clades? Like a single vaccine can be effective for against all the COVID clades like that.
Yeah. So I sort of mentioned before that clades have in many ways nothing to do with functionality on a larger sort of perspective, because clades are essentially genetic lineages which has arisen out of events which are essentially which have resulted in the emergence of a particular lineage, right? So in many ways they may not be functionally relevant, but having said that, there are exceptions to that. One of the exceptions is a D614G genetic mutation, which is a functional mutation in the spike protein, which is supposed to be involved in increasing the infectivity of the virus, not the mortality, not the lethality, but the infectivity of the virus. That means it can latch on much better to the human receptor and therefore are better infective. And one of the reasons suggested why the A2A clade has emerged and has sort of superseded all the other clades is probably because A2A clade has a D614G mutation, which has much higher infectivity. So there are functional events. Now, having said that, the vaccine per se, in many ways, the vaccines that are either in clinical trial or at least approved in some parts of the world are either reliant on the spike protein or spike plus envelope protein or whole virus. So in many ways, what is immunogenic and what is really important to elicit an immune response is essentially the exposed proteins of the virus, which spike predominates, and therefore the efficacy of the vaccine is mostly reliant on the spike protein, right? The clades and the genetic lineages are not typically because of mutations in the spike protein, but these are typically mutations elsewhere. Having said that, there are exceptions. The exception that I sort of mentioned is the D614G mutation, which is on the spike protein.
Thank you, sir. Thank you very much. And for the participants, we will take rest of the questions after sir's next session. So, sir, we can start the next.
Sure. Okay, can you see the browser?
No, sir, not yet.
Do you see this web page?
No, we don't. No screen is visible.
I think some error occurred in just a minute. I'll just try it once more.
So, for participants also, please give us 2 minutes to prepare. We'll start the session soon. We'll start the session soon.
Okay, so this is the web interface to the resources that we are sort of put together. Can you see the URL for the resource?
Yes, sir.
Yeah. Now, this web resource has two sections. One is the technical documents, and the other one is the web resources. And the technical documents essentially detail the things that I sort of mentioned before. One is the protocol for assembly and analysis of SARS-CoV-2 genomes. So this is a detailed step-by-step protocol. And anybody with a 4 GB RAM machine, Windows, any versions of Windows or Linux. For people who have Windows, you need to install Linux as a virtual machine. But anybody who has Linux with a 4 GB RAM machine should be able to follow this entire protocol. And this is mentioned point by point, each of these steps with each of these tools, and in a stepwise direction on how to install them, how to run them, and how to interpret the reports. So there's an extensive manual with all the commands that you would ever require to assemble a genome on a Linux machine. And of course, many of these datasets are publicly available on NCBI. So you should be able to go back and look at them. And there's a similar protocol which is available. Sorry for that. Similar protocol which is available for the phylogenetic clustering. Once you have the assembled genomes, you can go back and sort of systematically follow this protocol. Most of these tools are installable on Linux and very limited resources. We suggest around 4 GB of RAM, and the entire set of commands to install all the required tools and run a phylogenetic analysis and interpret the phylogenetic analysis and also visualize them is available on this. So you can also create a visualization. I'll sort of touch upon this visualization on our web interface. Visualization to interpret the phylogenetic tree is also available on this protocol. So anybody who is interested in learning about assembly and also learning about phylogenetic analysis can actually go back to these protocols and analyze them. Now, the second part of this is the web resources. There are a number of web resources which are available. For the time being, let me first introduce you to this web resource, which is called PhyloVis. Now, PhyloVis is a visualization interface for the SARS-CoV-2 genomes from India. We also have a Middle Eastern build. I will not delve deep into that. But for the time being, let me describe what is available for India and how you can go back and visualize this information. It takes a bit of time to load, so please bear with me. And all the methodology on how we created this resource is available in this link here called methods. You could also find more information of the clade I-A3I in this link in here. I'm sorry, this takes a bit of time. While it loads, let me also introduce the second resource, which is called Genomepedia. Genomepedia, as I stated, is essentially a search engine. You can search by particular strain name or a lab ID or a host or a country. For example, you can search for Australia and click on the search button. It will search, retrieve, and give you back all the virus isolates from Australia. You can put the drop down, and you can also understand where did this come from, what is the ID for this, and you can click and retrieve the information for this virus from the original website which hosts this virus. Apart from that, you could, for example, search for India. You could identify all the viruses which are in India. This is the virus, for example, deposit from NIMHANS. you could click in here and go to GISAID and sort of download this information. So that is Genomepedia. COVID beacon, I will come to back a bit later. Before that, I'll introduce this resource called INDICOV. Now, INDICOV is a resource which catalogs all genetic variations in SARS-CoV isolates from India. As I stated, there are around 4,000 odd isolates, and we have around 5,500 odd genetic variations which are available in this 4,000 odd genomes. The variants can be checked in Indian genomes as well as global genomes. The global genomes have much more number of genetic variations. I will not delve deep into that. And you could search by either the nucleotide position. You need to give the nucleotide position with a colon followed by a reference nucleotide and the alternate nucleotide. Or you could search by gene name. So for example, if you want to look at all S or the spike protein genetic mutations in India, you can just click here. And it pulls a drop down. It tells you what is the position of the genetic mutation, what is the reference, and what is the alternate. You can click on the gen view details. It opens up extensive set of information for this genetic variation. It will tell you it's an exonic genetic mutation, it's a non-synonymous genetic mutation. It tells you the protein change is D614G. So this is the spike protein I sort of mentioned, variation I sort of mentioned. It gives you a list of pathogenicity scores by SIFT. It looks at the conservation scores across other coronavirus genomes. It gives you additional information about the strains on what are all the isolates which contain this particular genetic mutation in GISAID. So you can, this is an extensive list of all IDs. I'll delve deep into it. It also gives you the allele frequencies, or in other words, how frequent is this variation in genomes from different states. So you have across all the isolates, around 4,000 odd isolates, we know that it's around 87%. Now you have the breakup by each state. In Uttar Pradesh, it's around 20%, Karnataka, it's around 43%, so on and so forth. And looking at the global SARS-CoV-2 genomes, approximately around this data is for around 100,000 odd genomes, it is around 81%. It also tells you the domains, if there are domains. It also maps the epitopes, the B cell epitopes, T cell epitopes, so on and so forth. It maps the structural context of the genetic variation. It also tells you whether it is in some primer or probe binding site. It is in none of the primer and probe binding site. And of course, it also tells you some positions in the genome which are prone to have errors. So if they are in some of these sites, whether they're hypermutable or sequencing error sites or homoplastic sites, it sort of mentions that information. And as a time series, it also mentions how the proportion of the virus have actually gone up or gone down. So this is from January until October. The frequency of these genetic mutations in each of the months is sort of displayed in here. It will also give you the geographical location from which this virus or this frequency is sort of retrieved. Is sort of a pictorial representation of this information, which is already there in the allele frequency table. So for each information, this is available. So if you are essentially looking at a particular protein or a particular genetic variation, and you want to dive deep into understanding this genetic variation, this is essentially the resource that you could go back. You could also go back and get all the IDs of all the data sets in here. You can click on the acknowledgement table, which provides all the IDs, all the references, and all the links to where you can download each of these sequences independently. So that is INDICOV. And very recently, we have put together this new resource, which is called FaviCov. Now, Favicov is a compiled resource for functional genetic variations in SARS-CoV-2. A functional genetic variation means there is some functional attribute to that genetic variation. It could be associated with, for example, infectivity. It could be associated with, for example, an epitope or immune epitope gain or an immune epitope loss, or it could be associated with, for example, clinical severity of infection. Like, for example, there are ORF8 genetic deletions which are associated with less severe infection of SARS-CoV-2. So functional genetic variations are sort of marked in here. This is the same D614G genetic mutations. And all this information is compiled from literature. So this is the search output. You can click on view details and it will list you a host of genetic information about this. It tells this is in the surface glycoprotein. It links out to all the relevant databases. It tells it is the D614G genetic mutation. It gives you the CYP polyphen and other epitope scores. It tells you whether it is in one of the primer sites, homoplasmic sites, sequencing error sites, so on and so forth. It gives you the global allele frequency across the different continents: Africa, Asia, Australia, Europe, South America, North America, and the literature curated functional information on what this mutation does is also summarized in here in a particular sentence. So you could go back, and an extensive set of references from which this information was garnered is also sort of listed in here. Now, this information is today available for over around 400 genetic variations for which there is literature information. You can search using a nucleotide change, you can search using a gene name, or you can search using an amino acid change. If you are looking at some analysis pipelines where you are analyzing large number of genomes, you can download all the variants in one single shot, the variant position and the related reference information. It doesn't provide the other information because it needs to be linked out to multiple different resources, but all the genetic variations and the references are available in a single file for download. There's also an extensive manual on how to use this resource, and all these data sets and the genomes can also be downloaded from the acknowledgement table out here. So this is Favicov. Now, the other resource that I would want you to look at is what we call as the COVID beacon. Now, the COVID beacon is essentially a new concept altogether. The concept of COVID beacon is that you could have your genomic resource in your lab or in your site, and in some cases you don't want to share the genomes because of some restrictions. Of course, it could be ethical restrictions, it could be country-wide restrictions. Some countries would not allow their genomes to be shared, but nevertheless you want the genomes to be analyzed remotely. So what is restricted is the sharing of the genome sequence, but what in most cases people would want is to access the genetic variant and understand whether there is a genetic variant in the genomes that are restricted. So this is what we call as a COVID beacon. So beacon essentially exposes the genetic variations and a set of metadata from datasets across the globe, which is now searchable across these datasets. This resource was built in collaboration with the Dennis Boer lab at CSIR in Australia. And the idea is you could search for a genetic variant. It searches across all the exposed databases. And in this particular case, there are 3 databases. This is the Indian SARS-CoV-2 genomes, this is the GenBank COVID-2 genomes, and the National Genomics Data Center in China, which has exposed its genetic information. And this genetic mutation I sort of searched, it tells me what is the frequency, what is the total number of samples having this, and it will also tell you on a map what is the frequency of this genetic mutation in different parts of the world. And like in Indicov, it will also tell you the month-wise frequency of genetic variation. This is a D614G. As you see here, it is almost 99% of all the isolates, for example, sequenced across the world. So this is the COVID beacon. If you have a genetic variation and you want to know more about it from multiple different resources in one single shot, this is probably the resource that you need to go back and look at. The phylogenetic tree is still loading, so I'll need to go back and check what really went wrong. But until then, I can take questions.
Thank you, sir. So, sir, we have two more questions from the participants. First is from Mr. Rajan. He asked that, like, often infected COVID-19 patients get negative PCR results. So, like, what is the reason? Like, why is it happening that so many people are getting negative PCR results?
So the question is, why are people getting negative PCR results? Is that
Yes,
the question?
infected people, sir, infected COVID-19 patients.
Right. So uh, a negative PCR is essentially a false negative report. Right? That means that an individual is infected, but his PCR is negative. Right? Now, that's exactly the point that I sort of stated in the diagnostic essay, that in approximately 1% of individuals in India, or 1% of viruses in India, they have genetic mutations in the primer and probe binding sites for major primer and probes that are used in kits. Now, it's a factor and dependent upon the kit that you use, because we know what kits have what primers and therefore are likely to be less effective. So in most cases, at least some of them are genomically confirmed and also available in literature now. Most of these cases could be because there is a genetic mutation in the primer and probe binding site, which doesn't now amplify the virus and therefore gives you a negative report, though you are positive for the virus. It's dependent upon the kit rather than the PCR per se. So it is always worthwhile to go back and look at the kit and understand whether they have a genetic variation which is mapping to the primer and probe site.
Thank you, sir. So, another question which like participants are like very curious to know that when you collect these kind of data like for such bioinfo analysis like the samples from Kerala, so how much time period does it take for collecting these data? What are the like criteria one should follow for such samples to be collected and for such bioinfo analysis? So,
Okay. So in Kerala, what we collected was essentially from one tertiary care hospital in North India, North Kerala, which caters to samples and population from adjoining 3 to 4 districts, because it was a major center across North Kerala. What we took was around 200 samples of adequate viral load and typically spaced across the previous 1.5 months of the outbreak. Ideally, you should take a random sample which is representative of the population, but in many cases that is not really possible. But near random sampling is what you typically do, so that you sort of have an idea of the spread of the virus and also have representation from individual outbreaks. Now we are actually extending this analysis to all the 14 districts in Kerala. There is a Kerala government program to understand the genetic epidemiology of virus, and we are also talking to many similar states across the country where the state has come forth to understand what is the genetic epidemiology of the virus and also use this information to understand where did outbreaks occur, how do you build in policy to curb the outbreak and also validate some of the policies. Like, for example, in Kerala, the extensive sort of quarantine measures put in place for the international travelers is sort of validating the fact that it was a proper procedure, and most of the outbreaks that has happened in the state has happened from travelers who travel from across the state rather from international travelers. So there are many approaches where you can sort of validate the policies that are put in place.
Thank you so much, sir. Sir, we have one last question. It's about the Linux, and like participants would like to know that is it necessary to use Linux in VirtualBox, or like Windows subsystem for Linux would work?
No, so you need to have a Linux implementation. That's all what I stated. You could have it on a Linux box. You could have it as fully Linux. It doesn't really matter. What only matters is that it needs to have a Linux environment, and you need to allocate enough resources for the Linux environment.
Okay, okay. Thank you so much, sir. So these were the questions, and thank you for answering them. And so one last question, sir, like if somebody wants to contact you or for some query or like for general information, so how can they connect with you?
You
So
could you could just email me.
yes.
That's the easiest way.
Okay, sir. So your email ID is available on the CSIR-IGIB web
Yes,
page.
yes.
Participants, anybody who is interested to talk to sir personally or about any query or information you would like to ask him, then you can ask sir via email, which is available on sir CSIR-IGIB webpage. So thank you very much. Thank you so much, sir. So that's for the next for this session, and our next session will be at 2 p.m. IST. Thank you, everyone.
Thank you
Uh,
very much.
Vinod, are you done? So we take your leave.
Sure.
Is it? Yeah. Thank you very much. Thank you so
Thank
much.
you.
Hope you all enjoyed the session. We'll get you back at Neha. When is the next session?
Mama,
Yes. So we resume at 2 p.m. IST. Thank you.